dear students, I welcome you all. Today we are going to start with the new chapter in physiology, and that is respiration in plants. So first of all, we are going to see what is the difference between respiration and breathing. So many students think that both of them they are same, but that's not the scenario. देखो breathing क्या है? Breathing is taking in of oxygen and giving out of carbon dioxide. मतलब inhalation and then exhalation. तो ऑक्सीजन अंदर आना, कार्बन डाइऑक्साइड बाहर जाना, ये जो सिंपल प्रोसेस है, दिस इज नोन एज ब्रीथिंग. सो लुक एट दिस डेफिनेशन हियर. यस, इट इज gaseous exchange, that is intake of oxygen and release of carbon dioxide. But what is respiration? क्या होता है? In respiration, actually this oxygen is going to be used for the breakdown of organic compounds, for breakdown of food. So respiration के अंदर actually this oxygen is going to be used for the chemical breakdown of organic compounds like carbohydrate, fats, proteins. So in all these compounds there are carbon-carbon bonds. Carbon-carbon bonds है, इनका breakdown होगा, oxidation होगा, and there is going to be release of lots of energy. तो यहाँ पे हमने क्या याद करना है? रेस्पिरेशन क्या है? रेस्पिरेशन इज नॉट जस्ट टेकिंग इन एंड गिविंग आउट. इट इज नॉट दैट. इट इन्वॉल्व्स ब्रेकडाउन. Very well aware of ATP. It is cell का पैसा. It is known as cellular currency of cell. Now, what are the respiratory substrates? तो कौन-कौन से substrate है? For कौन-कौन से substrates हैं which will undergo breakdown and there is going to be release of energy. So these substrates can be carbohydrate, right? Proteins, fats, and organic acids. So many diverse substrates are there. So many diverse. So if a question is asked here that out of all these, which is the most preferred one? सबसे ज़्यादा कौन सा preferred है? Fat preferred है? Protein preferred है? कौन है? Most preferred substrate out of all this for the respiration is carbohydrate. ठीक है? Now why it is so? We are going to discuss in forthcoming part of this lecture. Now this respiration, you know, it is a slow process. It's a sequential process. Stepwise है. मतलब एकदम serial order में चलने वाला है कि किसके बाद क्या बनने वाला है. Okay? So it is a slow process. So this one, it is stepwise process which consists of number of chemical reactions which are controlled by enzymes. So there is going to be glycolysis, link, Krebs, so many, and they are going to occur one after the another. Okay? Then, now energy released in these steps is going to be utilized for the formation of ATP. ऐसा नहीं है कि energy release हुई and immediately it is going to be used. No, this energy is going to be trapped in the form of small packets, small chemicals, and these chemicals they are ATP. So it is not like that all the energy produced after breakdown is trapped in the form of ATP. नहीं, ऐसा नहीं है. Now some amount is also going to be released in the form of heat. तो ये याद रखना है that energy released during the breakdown either it is trapped in the form of ATP or it is released in the form of heat. Okay, and generally students they think that majority of energy is trapped in the form of ATP, but that is not the scenario. Only 45% is trapped in the form of ATP and rest is released in the form of heat. तो हमें ये आपको ऐसे भी महसूस करते हैं कि जब आप खाना खाते हैं, खाना खाने के बाद थोड़ी देर के बाद आपको गर्मी लगती है, So from where this energy is coming? This energy is coming due to the breakdown of organic compounds. Okay. Now ATP is broken down whenever and wherever energy needs to be utilized. अब आपने ये छोटे-छोटे पैकेट्स बना लिए energy के, ATP के. Now these ATP molecules, they are going to be utilized wherever, whenever they are required in the cell. Okay, that's why what do you call ATP as? ATP is known as energy currency or energy packets of the cell. पैकेट्स है तो देखो जरा डायग्राम देखो एनर्जी इसका एटीपी का. लुक एट दिस एटीपी. सो इट्स फुल फॉर्म इज एडिनोसिन ट्राइफॉस्फेट. ओके, सो व्हेन वी से एडिनोसिन इट मींस इट हैज अ nitrogenous base adenine. You can look at this. This is nitrogenous base adenine. It has 2 rings, so it is a double ringed structure. Along with that, there is presence of the sugar, that is a ribose sugar. When we say ribose, it is a 5 carbon sugar. So ये हो गया base, ये हो गई sugar, दोनों को मिलाकर क्या बोलना है आपने? Adenosine. Fine. And along with that, there are 3 phosphates. So these 3 phosphates, they are arranged in series. Now this bond through which these 2 phosphates they are attached, you can actually represent it like this. This is how you represent high energy bond. This is also having ample amount of energy, so you can also represent it like this, but this is having maximum energy. Now this bond, when it will undergo breakdown, देखो जैसे तोड़ दिया, so this phosphate is going to be released and the energy released is 7.3 kilocalories. तो एक bond को तोड़ेंगे तो इतनी energy निकलेगी 7.3 kilocalories. Alright. Now, so before starting with that main steps, sequential order, glycolysis, link, Krebs, so before that we have to look at these important points which are there in the NCERT. Do plants breathe? Do they show a gaseous exchange? Yes, they show. But for the gaseous exchange, they don't have respiratory system. They don't have that complex respiratory organs like lungs. They don't have that. Yes, they have very simple structure for the gaseous exchange, and these structures are, one is this one, stomata, and another, these lens shape apertures. Please see here. These are lens shape apertures because they are lens shaped. So that's why you call them as lenticels. ठीक है, तो gaseous exchange is going to be there with the help of these pores. One is this one, that is stomata. Another one is this one, that is lenticel. Okay, so complex organs नहीं है, lungs वगैरह नहीं है. Okay, so simple structures, they are going to serve the purpose. Why it is so? Why it is so? Because they don't have great need for gaseous exchange. उनकी इतनी demand ही नहीं है. Demand is less. Demand for gaseous exchange is less. So I'm going to put an arrow down like this. इतनी demand नहीं है जितनी हमारी demand है, इनकी कम demand है. Okay. But sometimes great demands can be there, like during photosynthesis. During photosynthesis, great demand is there, and that can be easily solved with the help of the pores, stomata present on the leaf surface. So that can be easily managed. So during photosynthesis, ये सबसे एंड में यूज होता है. वाई सबसे एंड में यूज होता है? क्योंकि प्रोटीन एज अ रिजर्व फूड जर्नली होता ही नहीं है. प्रोटीन एज अ रिजर्व फूड जर्नली प्रेजेंट नहीं होता. मतलब कुछ केसेस होंगे पर जर्नली नहीं होता. ओके, सो व्हेन लेट्स से इन द प्लांट नो कार्बोहाइड्रेट इज प्रेजेंट, सो प्लांट इज गोइंग टू यूज फैट. ओके, let's say no fat is present, so now there will be turn of protein, but protein as a reserve food is not present now. So from where this protein is going to be used? This protein is present in the protoplasm. प्रोटोप्लाज्मिक प्रोटीन यूज होने लग जाएगा रेस्पिरेशन के लिए. मतलब protoplasmic enzymes, protoplasmic structural proteins, they are going to be used for the respiration. Means protoplasm तो खत्म होने लग जाएगा ना, protoplasm खत्म होने लग जाएगा, enzymes खत्म होने लग जाएगा. So when protoplasmic respiration will start, this is sign of unhealthy respiration, and ultimate fate after this respiration is death of cell. Protoplasm ही नहीं रहेगा तो death ही तो होगी, ठीक है ना. So this is unhealthy respiration because it is going to lead to consumption of protoplasm. इसके अलावा when protein they undergo breakdown, they form amino acid. Amino acids, they form ammonia. तो देखो protein breakdown will yield amino acid, and amino acid they are going to release ammonia, and we know this very well that ammonia is toxic. It is not only toxic for plants, it is toxic for animals, microbes, all the living organism. Even so, it means we can say protein respiration is not good because it is going to release toxic substances, but such type of toxic substances, they are not released here. That's why carbohydrates and fats, they are preferred as compared to the protein for the purpose of respiration. तो ये clear हो गया दो तरह का respiration. Now, so now we have to start with the story. So whatever respiration it is, whatever organism it is, now first process which is going to occur in the respiration, that is glycolysis. कोई भी ले लो organism, मतलब बैक्टीरिया ले लो, yeast ले लो, कुछ भी ले लो, दही खा रहे हो ना, दही में बैक्टीरिया, कुछ भी ले लो. First step which will occur in the respiration, your first process, that is glycolysis. Okay. Now the term glycolysis is derived from two words, Greek word. One is glykos, which means sugar. Lysis means breakdown, splitting. Glycolysis, it was first discovered by these three scientists, Amden, Parnas and Mayerov. So that's why this pathway is known as EMP pathway. So this, all these names, they are there in the NCERT, so they should be on the fingertips. EMP pathway कहना भी इन तीन scientists ने दिया ये pathway. The question arises here, where it occurs? कहाँ पर होगा ये process? This process is going to occur in cytoplasm. In all the living organisms, it will occur in the cytoplasm. Okay, so look at this prokaryotic cell. In prokaryotic cell, we know that well-defined nucleus is not present, so that has been shown here. This is not well-defined one. But in eukaryote, you can see this well-defined nucleus. Now, this is cytoplasm. Here, this is cytoplasm, guys. This one. Here, this is cytoplasm. So, in the cytoplasm, glycolysis is going to occur. So, it means whether it is a simple cell like prokaryotic cell or complex cells like eukaryotic one, in all the living organism, now this first process is going to occur in cytoplasm. ठीक है ये बात clear हो गया? Okay. Now, definition देखो अब NCERT की definition है, याद रखनी है. Okay, glycolysis is partial oxidation of glucose to form two molecules of pyruvic acid. So you know that glucose is 6 carbon compound, guys. 6 carbon compound है. So it will undergo breakdown and it will form two molecules of pyruvic acid. Each molecule is 3 carbon. Okay. तो 6 carbon compound का breakdown होगा. So this is going to yield two molecules of 3 carbon pyruvic acid. And ATP भी बनेगा. ATP ही नहीं सिर्फ. Along with that, there will be also formation of NADH. Now, so from where this glucose is going to come? कैसे आएगा, मतलब ग्लूकोज जो ग्लाइकोलाइसिस के अंदर एंटर करेगा. इट कैन कम फ्रॉम योर टेबल शुगर. योर टेबल शुगर इज सुक्रोज. So this sucrose, it will undergo breakdown by this enzyme invertase. तो एक glucose बन जाएगा and one fructose. Okay, so and this can enter glycolysis. Now there can be breakdown of starch to form glucose. So starch is a polymer of glucose. तो उसका भी breakdown हो सकता है. So this is how glucose is going to be available for the process of glycolysis. Hey, this process, it involves 10 steps. Now in the first 5 steps, see here, first 5 step, ATP use होता है. Amazing, amazing or surprising, surprising. हमने तो ये सुना हुआ है कि that in the respiration there is breakdown of food and release of energy occurs. But please, please, in the first few steps there will be utilization of ATP. So that's why you call it as glucose activation phase. Okay. Then in the next step, ATP form hoga. Along with the ATP, there is formation of NADH. So in the second phase, we are going to form ATP. So that's why it is energy extraction phase, and you can also call it as a payoff phase. तो जैसे कोई business होता है ना, बिजनेस में हम पहले पैसा लगाते हैं, तो यहाँ पर पैसा लगा and in this phase पैसा हमने कमाया, so this is payoff phase. So how much ATP is utilized, how much is going to be formed, वो पहले ही याद हो जाना चाहिए. Why you will wait कि glycolysis खत्म हो and then you will learn. शुरू से याद करो भाई. Okay, so there will be utilization of 2 ATP. Fine. And formation of 4 ATP and 2 NADH. So if we talk about the net ATP utilization or net ATP production, it is going to be 2. क्योंकि 4 यहाँ पे बना है और 2 यहाँ पर यूज हुआ है. ठीक है. Now this pathway I have told you earlier also that this is universally occurring respiratory pathway, because this pathway is the one which is occurring in all the living organism. कोई भी हो चाहे, मैंने बताया ना, बैक्टीरिया हो, yeast हो, plant हो, animal हो, कुछ भी हो. This is going to be common. This is known as universally occurring respiratory process. Okay, and where it occurs? Cytoplasm में होता है. ठीक है. So let's take up this question here, bacha. Now, in floating respiration, substrate used is or are. So we use in floating respiration carbohydrates and fats. So this is our right answer. Very easy question. Now let's begin our story of respiration, where I will be talking about glycolysis. 10 स्टेप पढ़ने हैं, साथ साथ ही याद हो जाएंगे, इसलिए टेंशन मत लेना. Okay, so we are going to start with the glucose. It is a 6 carbon compound. It is going to use 1 ATP. So from ATP, 1 phosphate will be added to glucose. So from this ATP, एक phosphate जो है वो glucose पे add हो जाएगा at the 6th position. तो 6th position पर add हो गया तो क्या कहेंगे आप उस compound को? You are going to call it as glucose 6 phosphate. Okay. So whenever there will be utilization of ATP or there is going to be formation of ATP, so there is utilization of enzyme which is known as kinase. तो याद ही रखो भाई, अब शुरू से याद रखो, जब भी ATP बनेगा या ATP use होगा, enzyme which is going to work at that step is kinase. Now here our substrate is glucose, क्योंकि substrate जो है वो glucose है. So that's why you are going to call it as glucokinase. Yes, so it's glucokinase. Now, whatever the enzymes are there, the enzymes, they are named like this only. They are named on the basis of their substrate. So, अगर यहाँ पे substrate होता fructose, so you would have said it as fructokinase. Fine. So, this is first step in the pathway. Now, moving on to the second step. So, now what is going to happen? Now, this glucose 6 phosphate, it is going to be changed into its isomer, and name of that isomer is fructose 6 phosphate. Because isomerization reaction is occurring, so simple name of enzyme is isomerase. And what is the complete name? Complete name क्या बोलेंगे आप? You are going to take substrate name also. You are going to add that name. So, it is going to be known as phosphoglucoisomerase. Okay. Now, coming to the third step. Now, third step पर एक और ATP use होगा. One more energy currency of the cell is going to be used. So, energy or ATP is going to be used only at the first and third step. Now, what will happen? It has got one phosphate at the 6th position. Now, one more is going to be added at the first position. तो एक तो छठे पे है ही, अब एक first पे add हो जाएगा. So, now what is going to be our product? Name of our product is going to be fructose 1,6 bisphosphate. An enzyme which is involved at this step, how you going to name it? Phospho, fructo. ATP use हो रहा है, so it is going to be kinase. This enzyme is very important enzyme, and in the short form it is written as PFK. So, why it is important, that we are going to study in the forthcoming part of this lecture or maybe next lecture कि इसका significance क्या है. Yes, it is basically like pacemaker. Pacemaker की तरह है. Pacemaker means कि कब glycolysis के rate को बढ़ाना है, when it needs to be slowed down. All this is under the control of this PFK. It is pacemaker enzyme. Pace बनाएगा भी कब बढ़ाना है, कब कम करना है. Okay. Now, coming to the fourth step. So, now what is going to happen? This substrate, it will undergo breakdown. This is going to undergo breakdown. Yes. After breakdown, there is going to be formation of two compounds, 3 carbon each. One you call it as DHAP. Just to remember it, DHAP. That is dihydroxy acetone phosphate. Okay. Then another compound which is formed here, that is 3-PGAL. Yes. So, that one is 3-PGAL. So, 3-PGAL is, full form is 3-phosphoglyceraldehyde. ठीक है. So, now these two compounds, they are isomer of each other. They are isomer. Now, only difference is that functional group in this first case, it is keto. तो बना देते हैं यहाँ पे भी keto है. Okay. And in the second case, functional group is aldehyde. So these are the isomers, okay. One is keto and another is aldehyde. So change in functional group is there. Enzyme which is involved at this step is aldolase. Yes, because they are isomers of each other. So enzyme which would be involved, that is again isomerase. Isomerase hai. And here these compounds, they are 3 carbons. So you can also use this word triose isomerase. So this is our enzyme here. So this is how we are done with the first phase of the cycle. In the first phase, we have used 2 ATPs, one at first step and another one at third step. And at the end of this phase, we have 2 PGAL. बहुत काम की चीज़ है भाई, बहुत काम की चीज़ है PGAL, because this is going to give us lots of energy, lots of energy. So now we will begin with the second phase. So in the second phase, now kahani, now story is going to begin with that PGL. And how many PGLs you have formed? You have formed 2 PGLs here. So let's first see what is going to happen with the one molecule of PGL. So nobody is going to say that these are the 3 molecules. Why I have written 3 here? Because phosphate is attached at the third position. तीसरी position पर add है, तब लिखा है P, यहाँ पे 3P. So you should not be thinking that it represents 3 molecules of PGL. नहीं, ठीक. Now this PGL, it undergoes oxidation. Respiration का मतलब ही oxidation होता है भाई. So this is 6th step. This is 6th step. So if a question is asked here, at which step first oxidation reaction is going to occur? Respiration का मतलब oxidation होता है, तो वो ये step होगा 6th step. And of which compound? PGAL. Now oxidation is going to yield hydrogen atoms. Hydrogen atoms. So there is going to be release of, सुनना जरा ध्यान से सुनना, there will be release of 2 hydrogen atoms from 1 PGL. एक PGL से 2 hydrogen atoms release होंगे. 2 hydrogen atom means 2 hydrogen ion and 2 electrons. Now what is going to happen to these hydrogen ions and electrons? These are going to be taken up by NAD+. So NAD+ can take only 2 electrons from here. 2 ले लेगा यहाँ से और 1 hydrogen ले लेगा यहाँ से, बस 1 hydrogen ion. So it would be changed into NADH. दोबारा सुन लीजिए. It is going to take 2 electrons यहाँ से, 1 hydrogen ion from here. So it is going to form NADH and 1 hydrogen ion is extra. You are going to put it like this. ऐसे. Fine. NADH plus H plus. This is what is going to be formed. तो याद रखना है. 1 PGL is going to yield 2 hydrogen atom, but that is going to form only 1 NADH. एक. तो ये एक molecule, ये 2 atoms, and this is going to be 1 molecule. Okay. Now at this step, even there is addition of water. There is addition of phosphate. Okay, and keep this in mind that this phosphate is not coming from ATP, it is not coming from ATP. So now what is going to be our compound? Resultant compound is going to be, so 1 phosphate is already present at third position. So 1 will be added at first position. So compound is going to be 1, 3, 1, 3, base, phospho, 2 हो गए ना, तो base phosphoglyceric acid. Yes, aldehydes. Listen to this. Aldehyde after oxidation results in the formation of acid. तो अब aldehyde नहीं लिखना है. Now aldehyde has been changed into acid, and what is the name of this acid? In the short form you can call it as BPGA. In the short form you can call it as BPGA. Okay. So because we have two molecules of PGAL, so you can multiply everything by 2. So 2 PGAL. So 2 water molecules going to be used, 2 inorganic phosphate. So there are going to be 4 hydrogen atoms multiplied by 2, then 2 NADH are going to be formed, and 2 BPGA. So we multiplied everything by 2. तो ये जो 6th step है यहाँ पे बहुत कुछ हो गया, बहुत important है. NADH formation is there, okay. Water addition, inorganic phosphate is added. Now what is going to happen to this BPGA? Okay, I think you can tell me the name of enzyme which is involved at this step. So many times I have told you in the physiology, whenever there is going to be formation of NADH or there is going to be utilization, enzymes which are involved, they are known as dehydrogenases. Okay, bacha? So here dehydrogenase enzyme is involved. So what is going to be the complete name of the enzyme? You can call it as phosphoglyceraldehyde dehydrogenase. So name of enzyme is on the basis of the name of substrate. Fine. अब देखो. Now from this PGA, now phosphate which is present at the first position, this is going to be removed. देखो देखो देखो, अभी लगाया था, अभी remove कर रहे हैं इसको, ठीक है, पर ऐसे नहीं, अब इससे पैसा बनेगा, ATP formation. So this phosphate which is removed here, which has been removed here, it is going to join with the ADP. Hey, this one is contributed by bis. Yes, it is going to join with the ADP. There is formation of ATP. So I have told you now, whenever there is going to be formation of ATP, enzyme used is going to be kinase. And if you have to mention the complete name, this is going to be bis-phosphoglycerate kinase. You can call it as kinase, okay. So because we have 2 BPGA, so this is going to yield 2 ATP. So which compound is left now? We are left with 3, यहाँ से ले लो भाई, 3 phosphoglyceric acid. This is our resultant compound, 2 molecules. Okay. So at this step, that is the 7th step. 7th step का मतलब हो गया James Bond step. At the 7th step, something special has occurred. That is, there is formation of 2 molecules of ATP. Fine. And this ATP formation which has occurred here, it is direct formation. Direct का मतलब क्या हुआ? You might be knowing this, or we have learned this actually, we have learned this in the photosynthesis, that ATP formation can occur with the help of F0F1 complex. F0F1 complex can form ATP, but here we are forming ATP directly. We are not using any F0F1 complex. So when there is direct ATP formation, उसके लिए एक नाम होता है भाई, इसको कहना है आपने SLP. SLP full form, substrate level phosphorylation. ठीक है, तो ये हो गया हमारा जेम्स बॉन्ड स्टेप. And at the James Bond step, we have done something very special. We have formed 2 ATP. और इसके ऊपर वाले step पर we have formed 2 NADH. They are also important. Now coming to the 8th step. See here. Now at the 8th step, very simple. At the 8th step, now 3 PGA is going to be changed into 2 PGA. तो मतलब सिर्फ shift होगा carbon का, मतलब phosphate is going to change its position with respect to carbon. So now it is going to be changed into 2 phosphoglyceric acid. Right, 2 molecules. An enzyme which is involved here, that is mutase. Can we say mutase is a type of isomerase? क्या कहते हैं? Is it a type of isomerase? Yes, because here only we have changed the position from third to second, so these are isomers only. Mutase is type of isomerase. Okay, guys. Now coming to the 9th step. At the 9th step, now there is going to be loss of water. Okay, dehydration reaction is there. So loss of water is there because we have 2 molecules. So you have to show loss of 2 water molecules here. And what is the resultant compound? It's our jana pehchana PEP. Jana pehchana, that is phosphoenol pyruvate. Phosphoenol pyruvate. Enzyme involved at this step is enolase. Then coming to the last step. 10 steps hai na total. Last step. So what is going to happen at this step? इसकी full form क्या है? Phosphoenol pyruvate. So it has one more phosphate. One more phosphate is left in this compound. तो हमने किसी को भी waste नहीं जाने देना. We are going to yield ATP from that. So phosphate is going to be removed, and ultimately we are left with 2 molecules of pyruvic acid. देख लो ना, phosphoenol pyruvate. तो phosphate remove हो जाएगा, pyruvate, we are left with 2 molecules of pyruvic acid. Now this phosphate is going to be used for the formation of ATP in the similar manner which we have discussed here. SLP1. Phosphate यहाँ से release होगा. It is going to join with the ADP, and there is going to be formation of ATP. So what do you say? How many molecules will be formed? क्या लगता है कितने molecules बनेंगे? Because you have 2 molecules of PEP, so surely it is going to yield 2 molecules of ATP here. And enzyme कौन सा है? Enzyme, it's kinase. Full name is pyruvate kinase. ये generally जो enzyme का नाम होता है, that is on the name of substrate. But here you can see that name of enzyme is on the basis of product. So sometimes that also happens, that is there. But जो generally enzyme का नाम होता है, that is on the basis of the substrate. Okay? So we're done with the majority of part of glycolysis and what we have seen in the second phase. So let me scroll up a bit, that here we have formed 2 NADH here. They are very important, 2 NADH, very important. And here we have formed 2 ATP, सीधा सीधा. So सीधा सीधा is substrate level phosphorylation. ऐसे याद करो, ऐसे सीधा, ऐसे substrate level. Then at the last step, even we have formed 2 ATP सीधा सीधा. So total we have formed 4 ATP through SLP. Right बच्चा? Now, अगर ऐसा question हो, if question is like this, okay, let me change the page. If the question is like this, that from 2 molecules of glucose, there is formation of how many molecules of PGAL, then what do you want to say? कितने बनेंगे? From one molecule of glucose, there is formation of 2 molecules of PGL. अभी पढ़ाया 2 molecules. So if we had 2 molecules of PGL, so you will see 4 molecules. So if 2 molecules of glucose are there, so there will be formation of 4 molecules of PGL. And PGL is बहुत important because इसी से hydrogen निकलेगा, hydrogen, and that is going to be taken up by NAD plus to form NADH. Actually कहानी सारी hydrogen की है. How this hydrogen is useful? What it is going to do? That we are going to study in this respiration in the next lecture. Now from 4 molecule of PGL, how many redox equivalents will be released? Socho. From 4 molecules of PGL, how many redox equivalents will be released? Redox equivalents means hydrogen atoms. Hydrogen atom ये मतलब, हाँ जी, redox equivalents कह सकते हो. So we can write here. So how many redox equivalents will be released, and they are nothing but hydrogen atoms. From one PGL, okay, there is a release of 2 hydrogen atoms. And if we have 4 PGL, so 4 into 2, 8 hydrogen atoms will be released. Okay? So these are the questions which are generally being asked from this part. 8. दोबारा बताऊं 8 कैसे हुआ? Yes, from one PGL, I have told you 2 hydrogen atom release होंगे. And if you have 4 molecules, so 4 into 2, 8 hydrogen atom. Now these 8 hydrogen atom, they are going to form how many NADH? कितना NADH बनेगा भाई, कितना? 2 hydrogen atom, they are going to be used for forming 1 NADH. So 8 hydrogen atom, they are going to yield 4 NADH. समझ आया कि नहीं आया? Clear हुआ? Okay, so what is the another name for hydrogen atom? You can call them as redox equivalents. Okay? So whatever food you are eating, whether you are eating carbohydrate, fats, proteins, you are eating food for hydrogen atom. You know? So these hydrogen atoms, they are going to come out. ये देखो जैसे बाहर आ रहे हैं, ये बाहर आ रहे हैं, तो they are going to be taken up by NAD plus, forming NADH. And this NADH is going to enter ETS. ETS में enter करेगा इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में, और एक NADH में इतनी एनर्जी है कि 3 ATP बना सकता है. तो दोबारा सीक्वेंस सुना. Whatever you are eating, pizza, burger, dosa, sambar, idli, whatever, that is for hydrogen. Now once hydrogen atoms they are removed, they are taken up by this NADH. NADH is going to enter ETS, and एक NADH से 3 ATP बनेगा. So it's all about hydrogen ion. It's all about hydrogen ion. Whatever you are eating, that is just for hydrogen atoms. Clear है ये point? Everyone is clear? So if we revise the glycolysis, so what we can see here, that our very first step is this one. Glucose 6-phosphate has been formed. It has been changed into fructose 6-phosphate. So पहले glucose 6-phosphate, fructose 6-phosphate, then fructose 1,6-bisphosphate. Okay. Then टूटा lysis हुआ, lysis. तो lysis से क्या बन गया? एक ढप बन गया और एक PGL बन गया. Now ढप has also been changed into PGL. So you are having how many molecules? How many molecules of PGL? 2. And these PGLs are very important because they are going to produce hydrogen atoms. Now this PGL is changed into 1,3-phosphoglyceric acid, BPGA. BPGA से PGA 3, फिर 2 PGA, फिर अपना जाना पहचाना PEP, and then ultimately 2 molecules of pyruvic acid. So it means that in glycolysis there is not complete breakdown of glucose. Complete होता तो ये नहीं कहते कि टूट गया glucose और carbon dioxide और water बन गया. नहीं, there is partial breakdown, which releases 2-pyruvic acid, which forms 2-pyruvic acid. Along with that, there is formation of ATP and NADPH. So glycolysis is not going to release all the energy which is present in glucose. It is said that it is going to release only, James Bond वाला number कौन सा है हमारा? 7. Yes, it is only going to release less than 7% of energy which is present in the glucose. याद रखना है, line of NCERT. Okay, so this less than 7% of energy which is released, or not all of it is trapped in ATP. तो सबसे ATP नहीं बनेगा, because some of it is also going to be released in the form of heat. So this is very first step of respiration, very first step or very first process which is occurring in everyone. Bacteria ले लो, yeast ले लो, कोई भी ले लो, plant ले लो, animal ले लो, insect ले लो, कुछ भी ले लो. This is very first process. It is occurring in all the living organism, and we have discussed where it is occurring. It is occurring in cytoplasm. Everywhere it is occurring in cytoplasm. It means for this first process there is no requirement of mitochondria. तो कोई भी नहीं बोलेगा ये mitochondria में कुछ हो रहा है. Everything is occurring in cytoplasm. Nothing is occurring in mitochondria. Now this pyruvic acid which we have formed here, अब इसका होगा क्या? It has 3 fates. 3 fate है इसके. Now this pyruvic acid, it can be changed into alcohol. This is alcoholic fermentation. It may be changed into lactic acid, then it's lactic acid fermentation. And then if oxygen is present, in the first 2 cases oxygen is absent, and if oxygen is present, it will undergo complete breakdown. पूरी तरह टूट जाएगा. Carbon dioxide, water, बहुत सारा पैसा बनेगा, ATP बनेगा. That is aerobic respiration. So this pyruvic acid, it has 3 fates that depends upon the cell requirement, then cellular conditions, that whether it is alcoholic, lactic acid बनेगा कि पूरा का पूरा टूट जाएगा. तो वो depend करेगा क्या cellular requirements है, क्या conditions है, उस पर depend करेगा. So what is going to happen to this pyruvic acid, that we are going to study in the next class. We are going to study how NADH is so special, how it is going to form 3 ATP. एक NADH से 3 ATP कैसे बनेगा? How it is going to enter the ETS? What are the complexes which are involved? ठीक है? I hope so. This lecture, this part of respiration is very much clear to all of you. So here we started off with the definition. Then we have discussed why plants don't require complex respiratory organs like lungs and all. Then I started off with this glycolysis. We have discussed it and even we have revised it. Okay. So in the next class, यहाँ तक तो याद कर लेना तुम. Okay. Then so that we can do questions from the previous part as well as the part which we are going to study in the next class. That's all about the today's lecture. See you. Bye-bye everyone.